Hey, so my name is Drew.
And I'm Austin Neeley.
This is the what's the name?
With the meta.
Meta. That's
This
right.
is week 12 with the meta. All right, screen sharing this.
There. It's on the left one. It's the left one. Oh wait, never mind. You're right. You're right.
Cool. All right, number one. So right here we just got a typical word bank. You know how these ones work. So which of the terms describes or is an example of a regulatory protein? Well, the repressor, the activator, and the cap. Those are all examples of regulatory proteins. Which of the following describes an example of a small effector molecule? Well, alactose, inducer, and cap are all. It should actually be. It should be. That's fine. Okay, so it'd be lowercase c, big A, big A. Whatever, doesn't matter. Which of the terms describes as an example of a sequence found in DNA? Well, +1 and -10, lac A and lac Z. Which the following is not involved in lac operon? That would be inducer, chem, and alactose. Which of the following is an example of a structural gene? Well, that would be that'd be lac A and lac Z. And it is your turn, Drew.
Okay, cool. So this is the fill in the blanks. Pretty much is in the notes. So the lac operon is regulated by two inducers, C-amp and sorry, I can't pronounce really well. Allolactose. The binding site for signaling factor in RNA polymerase is called LAC-P, while the operator site is the binding site for activator that regulates the lac operon. The enzyme adenylycalase converts ATP to CAMP. The -35, -10, +1 sequence are found within the LAC-P portion of the lac operon. The LAC-L gene encodes the lac repressor, while the LAC-Y gene encodes the lactose primase. The mutation can regulate the gene expression in bacteria by causing transcription to terminate prematurely. An E. coli strain that contains 2 copies of the lac operon is called a merozygote strain.
All right, perfect.
Oh shoot, my bad.
Oh,
shoot.
All right. All right. Okay, so 3. Suppose you're examining lac operon in E. coli and you experience the activity beta-galactoside is measured by the X. Okay. So I just need to remind myself between these ones. Okay, guys. All right. So for this problem, assume that your E. coli cells contain a wild-type lac operon components, and allactose is absent in all cases. The abundance of ATP in all cases. Okay, perfect. So if IPTG is absent, glucose is absent, and X-gal is present, then it will be normal. And that's just the way it is. I mean, the glucose is absent, then it's not being done first. We need the IPG to make it blue, and gal. Yeah, so it's fine. Okay, so the IPTG is present, glucose is absent, the X-gal is present. It's going to be blue because, well, the X-gal, we need that for it to be blue, and the IPTG is like what helps it be processed. C, the IPG is present, glucose is absent, X-gal is absent. Well, because there's no X-gal in there, it can't be blue because that's kind of the stuff that is blue. So that's easy. For D, that's IP. ET is present, glucose is present, G, yeah, X-gal is present. It's also normal because the glucose is actually being processed first before it even gets to the lactose that's there. The IPTG is present, glucose is present, and X-gal is absent. It's kind of same-ish. It's going to be normal because X-gal isn't there even though the IPT is present and the glucose is present. And that is that. You want me to do 4 or do you want to do 5?
I'll do 5.
I'll do 4. All right, so 4 is a pretty easy one if you just look at it. So letter A. So the pluses mean the wild type gene that they're working perfectly. I, so P stands for promoter. O stands for operator. Operator, you just need to know that. So if the I is plus, that means the, what's it called? Starts with R. Repressor, thank you. Repressor is being made, and it attaches on to the promoter and background. So that'll be inducible. We don't have the repressor, it's not going to be working here, so that's non-inducible. This one works because of the second strand right here, otherwise it wouldn't because our promoter is defective. I think we have this one right here that's making enough for it. So it's inducive. So it's being repressed, the operator, the promoter is all working. Now this, because we have this defective repressor, it attaches and won't let go. So that's non-inducible. Same thing kind of here, but it also happens right there. So that's constitutive. 4.
So for 5, we're looking for, let's see, we'll check. 0 in case you don't, we're looking for these genotypes will indicate no yellow color, amount yellow, or the 2 copies of light blood pressure that is transcribed. So for A, the E. coli genotype is, I can really see it, I minus, O plus, P minus. So if it's going to be P minus, right, then both the lactose and lactase will not produce any yellow color.
Right. Yeah. So
Okay.
already off the bat, so I can do anything.
Right away. Okay. And then if that's the case, then if P plus is the same is if P plus makes the lactose lactase go through, then they both have one yellow color. C, is that right? Right?
Mm-hmm, that's
Okay.
right.
So far.
Being repressed, it kind of just binds to the operators.
Okay. For C, pretty much the same thing. Both pluses. It's gonna be 2 on the lactose and 1 minus on the lactose. No, sorry, 2 on the lactose plus and 1 on the lactose minus. For D, pretty much the same thing. There's a plus on the P, so it'll be +1, and there's a minus on the P, so it'll be 0 on the lactose, negative. For E, how'd you explain this one?
Okay, so because it's not being repressed, it's just not going to be working out. But if you look at the second strand right here, it binds to it and like doesn't let go. So there's no lactose there. So we have a Z. Very 0 right there.
Both zeros.
Yeah. Okay. Perfect, and that is it. Thank you so much, everybody.
Cool.
Bye-bye.
See ya.